Polymerase chain reaction, i.e. PCR, was first patented by Carey Mullis. The central concept underlying PCR is to use short single strands of DNA molecules to enable the amplifying of targeted regions of DNAs. Before Mullis, such targeted specific tools in the study of DNAs had already been widely used. Mullis' main contribution was to make the scale of applications much larger by repeating the process over and over again, namely the chain reaction. The chain reaction is made possible because DNA molecules can be copied by adding essential components of DNA to the reaction pool with DNA polymerases to activate the synthesis. So, as long as the copies made from the previous cycles are still kept, the next time when this process is repeated, more copies can be made from the previous copies now serving as templates. This is an exponential process, so we can get many DNA copies in a short time. Quantity of product can be used to determine the initial quantity of the template DNAs. Typical steps of a PCR cycle include heating the DNAs to disentangle the double-stranded molecules, then cooling the reaction pool to enable the binding of specific short sequences, called primers, and the third step to enable the extension of DNA copies. One application requiring PCR as validation is the research involving alternative splicing. Alternative splicing refers to the theory proposed by Walter Gilbert that genes can be spliced and regions of a gene can be recombined to produce different matured RNAs from the same gene. The regions of a gene that can be translated into proteins are called exons, and the regions in between exons which are not expressed are called introns. However, recent research has revealed that introns also have regulatory functions in the cell. Alternative splicing is important because it can enable the complexities of products decoded from one single gene, allowing complicated functions in organisms. So, alternative splicing makes various forms of proteins with different functions from a single gene, and PCR is currently a reliable way to validate the data. We can't be as romantic as poets to assert a conclusion without rigorous evidence, and that's why data validation is such a big deal.